Right now, you can see a color that does not exist. Not a rare color, not an unusual color, a color with no physical reality whatsoever. Look at any magenta object, a flower petal, a neon sign, a phone case. What you are seeing has no corresponding wavelength of light. Stretch out the entire electromagnetic spectrum and you will find red, orange, yellow, green, blue, violet, but no magenta. The color exists only inside your brain. Your visual system invented it to solve a mathematical problem that the physical universe left unsolved. And magenta is not alone. An entire family of colors, every purple and pink between red and violet, is a neurological fabrication, a perceptual shortcut your brain runs automatically every time you open your eyes. You are not seeing reality, you are seeing your brain's reconstruction of it, filled with colors it invented because physics didn't provide them. Let's go in. Light is electromagnetic radiation, waves of oscillating electric and magnetic fields propagating through space at 300 million meters per second. The human eye can detect a narrow slice of this radiation, from wavelengths of roughly 380 to 700 nanometers. Within that range, different wavelengths produce different color experiences. 700 nanometers is red, 550 is green, 450 is blue, 380 is violet. This range is the visible spectrum, and it is a straight line. At one end, red, at the other, violet. Between them, every color the physical world can produce with a single wavelength of light. This is where the problem begins. The human eye contains 3 types of color-sensitive cells called cones, each tuned to a different range of wavelengths. The S cones respond best to short wavelengths, roughly 420 nanometers, what we call blue and violet. The M cones respond best to medium wavelengths, around 530 nanometers, what we call green. The L cones respond best to long wavelengths, around 560 nanometers, what we call yellow and red. This 3-receptor system is called trichromacy, and it was first proposed by Thomas Young in 1802. Young was a British polymath, physician, physicist, linguist, who worked out that 3 primary colors of light could reproduce any color the eye can see. Half a century later, Hermann von Helmholtz refined the theory and produced the first detailed account of how the 3 cone types work together. The Young-Helmholtz theory of trichromatic vision became the foundation of color science. You do not see wavelengths, you see the ratio of activation across your 3 cone types. Color is not a property of light, it is a property of your brain's response to light. Now here is where magenta appears. Red light at 700 nanometers drives the L cones hard. Violet light at 400 nanometers also triggers a secondary response in the L cones, because the L cone's sensitivity curve has a small uptick at very short wavelengths. So when your eye receives red and violet light simultaneously, both ends of the spectrum are active at once. The brain must produce a color experience from this combination, but it cannot average the wavelengths, because the mathematical average of 700 and 400 nanometers is 550, and 550 nanometers is green. Your brain would have to show you green every time you saw red and violet together. This is wrong. It is perceptually wrong, it is biologically wrong, and the brain rejects it. So instead, it invents a color. The brain takes the signal, high L cone, high S cone, low M cone, and maps it onto a perceptual experience that correctly distinguishes this combination from green. That experience is magenta. Magenta is the brain's solution to how to represent a mixture of spectral extremes without confusing it with the spectral middle. It has no wavelength because no wavelength produces it. It is an extra-spectral color, constructed entirely inside the nervous system. The key to understanding this is the shape of the color space the brain builds. The physical spectrum is a line, straight, finite, running from violet to red. But the perceptual color space the brain constructs is a circle, or more accurately, a wheel. In this wheel, red and violet sit almost next to each other, connected by the purples and magentas that have no counterpart on the spectrum. The wheel closes the gap that the spectrum leaves open. This is not arbitrary, it is the brain's way of representing the trichromatic signal space completely and without contradiction. Ewald Hering, a German physiologist working in the 1870s, proposed the opponent process theory of color vision, the idea that color is encoded as opposing pairs, red versus green, blue versus yellow, light versus dark. Hering's theory seemed to contradict Young-Helmholtz, but it was actually describing the next stage of processing, where the brain computes color differences rather than absolute cone activations. When both the red signal and the blue-violet signal are strong while the green signal is weak, the opponent channels report something that is neither red nor violet, but partway between, and that something must be magenta. The chromaticity diagram, developed by the International Commission on Illumination in 1931, makes this structure visible. It is a horseshoe-shaped map of all the colors a human observer can see, with spectral colors running along the curved edge. The straight line closing the bottom of the horseshoe, called the line of purples, contains every color that has no single wavelength counterpart. Magenta sits on this line. Every color on the line of purples is extraspectral. They are all, in exactly the same sense as magenta, invented by the brain. Semir Zeki at University College London has spent decades identifying the neural basis of color experience, focusing on area V4 in the visual cortex. His work, beginning in the 1970s, showed that V4 neurons respond to color in a way that is already opponent processed. They fire to specific hues, not to specific wavelengths. A V4 neuron tuned to red fires not just for 700 nanometer light, but for any stimulus the opponent system categorizes as red, including combinations of wavelengths that individually would not look red at all. The brain's color system is constructive from the very beginning of cortical processing. Color constancy deepens this further. A red apple under blue indoor lighting reflects very different wavelengths than a red apple in orange sunset light, yet the apple looks the same color in both conditions. The brain is actively correcting for the light source, computing the reflectance properties of surfaces rather than reporting raw wavelengths. Edwin Land, the inventor of Polaroid photography, demonstrated this in experiments in the 1970s. Land showed the brain can perceive a full range of colors from only 2 wavelength bands, as long as the scene contains enough reflectance variation to compute contrast. The color you see is not the color of the light hitting your eye. It is the color the brain has inferred the surface must be, given everything else in the scene. The internet made this viscerally clear in February 2015, when a photograph of a dress triggered a global disagreement. Half the population saw the dress as blue and black. The other half saw it as white and gold. The dress was objectively blue and black, but the photograph was ambiguously lit, and different observers' visual systems made different assumptions about the light source. Those who assumed warm lighting subtracted orange and saw blue. Those who assumed cool lighting subtracted blue and saw gold. Same image, same wavelengths, radically different colors. Because color is not in the image, it is in the brain's interpretation of the image. What does all of this mean for the nature of what you see? The philosopher Daniel Dennett argued that color experience is a paradigm case of perceptual construction, a model, not a direct readout of reality. You do not have access to the electromagnetic field around you. You have access to your brain's rendering of that field in a perceptual vocabulary shaped by evolution that only loosely maps onto physics. Magenta is not an error in this model, it is a feature, a creative solution to a representational challenge the physical world did not provide a ready answer for. Now here's the part that makes this personal. Every color you have ever seen is, in this sense, a construction. Red is not red because of some inherent redness in 700 nanometer light. It is red because that is what the L-cone dominant signal gets labeled in the perceptual vocabulary your visual cortex uses. A mantis shrimp, with 16 types of photoreceptors compared to your 3, lives in a color world so different that the two experiences share almost nothing. A dog, with only 2 cone types, lives in a world where red and green are indistinguishable. The colors you experience are not the colors of the world. They are your species' particular solution to encoding wavelength information with 3 detectors. And this applies far beyond color. Your entire perceptual world is built this way. Compressed, interpreted, filled in, constructed. Sound is not the compression waves in the air, it is your auditory cortex's rendering of them. Pain is not damage, it is your brain's alarm signal about potential damage, calibrated by context and expectation. Magenta makes this visible because it is so clear-cut. The wavelength is absent, the color is present. There is no other explanation, the brain made it. But here's what that does not mean. It does not mean that magenta is not real. It does not mean your experience of color is a mistake, or that the world is less vivid than it appears. The philosopher Thomas Nagel asked, "What is it like to be a bat?" A bat navigates by echolocation, perceiving a world of acoustic shapes and distances that has no analog in human experience. Nagel's point was that there is something it is like, a subjective quality, to inhabit any sensory system. Magenta is part of what it is like to be you. The electromagnetic spectrum does not contain it, but your experience does, and your experience is as real as anything in physics. The brain did not lie when it invented magenta. It solved a problem. It needed a way to distinguish red plus violet stimulation from green, and it found one. That the solution required creating something with no wavelength is not a flaw. It is a sign of how creative biological systems are when physics leaves a gap. Every sunset you have ever found beautiful was partly built by your visual cortex. Every flower whose color stopped you was partly your brain's contribution to the scene. The world is not less beautiful for being constructed. It may be more beautiful, because the construction is yours. The color that has no wavelength is the color your mind built to bridge the gap where physics ran out. And every time you see it, you are seeing proof that you are not a passive receiver of the world. You are one of its co-authors.